Hello, this is Mr. Arthur, and we're going to look at Biology, Higher Human Biology Unit 1, starting with the first key area, which is about cell differentiation and about cell division. Okay, so at the end of this video, hopefully you can state the qualities that make a cell either somatic or germline. There's two types of cell, or two ways we're going to group our cells. You should be able to describe the difference between mitosis and meiosis as different types of cell division. You have to state what is meant by the process cellular differentiation. And you have to describe different therapeutic uses of stem cells. And you have to describe why cancer develops and what leads to the spread of cancer. So let's start with somatic cells. Basically, these are pretty much all of the diploid cells in your body, apart from your two gamete cells. So that's the haploid sperm and the haploid ova, as well as the stem cells which divide to create them, okay? So sperm, ova, and the stem cells which make them aside, so that's the cells involved in reproduction, every other cell in your body is diploid. It has 46 chromosomes, and we call these somatic cells. Now your somatic cells divide by mitosis to form more somatic cells, okay? So remember, mitosis maintains the chromosome complement. That means the cells remain diploid after cell division. Now, germline cells, if we look at the DNA here, they're lined up as the 23 pairs of chromosomes. Each pair is called a homologous pair of chromosomes. So germline cells are our gametes, that's our sex cells, the sperm and the egg, the ova, and the stem cells which divide to form these gametes. So this is what I mean by germline cells. The germline cells divide by both mitosis, when they want to maintain a diploid chromosome complement, and by another type of cell division called meiosis. Meiosis ends up with haploid cells, so the chromosome complement is not maintained in meiosis. In fact, that's how you end up losing one set, so you have a haploid sex cell. So let's look at mitosis. The nucleus of a germline stem cell can divide by mitosis to maintain its diploid chromosome number. So this means if the germline stem cell divides, it makes two more identical germline stem cells, which is important so we don't run out of germline stem cells. It's diploid, so it has 23 pairs of these homologous chromosomes. Now, this second type of cell division is new to you. This is called meiosis. The nucleus of a stem cell can divide by meiosis, and if it does that instead of mitosis, it has two stages to the division. Step one, first of all, the homologous chromosomes, as shown, separate. And then secondly, the chromatids separate. That means you end up with a haploid gamete which contains a single set of 23 chromosomes. Now at National 5 we use the word chromatids slightly differently, so it can be a little bit confusing, okay? But just try and memorise this. It's homologous chromosomes in the first step, and then the chromatids separate in the second step. And that means that you end up with a haploid cell at the end. So this quick video shows you mitosis, the two daughter cells which are identical, and then meiosis, which end up with the first stage when they split, but then it splits again, this time with the chromatid splitting, so you end up with 4 haploid gamete cells at the end. Okay, you can pause this video now and have a look at the difference between the two. Remember, if you're asked to compare them, make sure you mention both mitosis and meiosis. Now, germline cells can use either type of cell division. Somatic cells only use mitosis. Again, you can pause the video on this. This is the SQA course notes, what is going to be in the exam. This is what you need to know about this topic, and then press play when you're ready to continue. Okay, so what we're looking here is a stem cell in the middle, and the stem cell is an unspecialised cell. That's why we just draw it as a basic circle. All the cells on the outside, these are specialised cells. We call them specialised cells because they have a special function in the body, and because they have a specific function, they have usually a special shape. So muscle cells at the top are really long and they're able to contract. Red blood cells in the bottom right have that biconcave shape, you'll know from National 5, so they're more efficient at absorbing oxygen. The immune cell, if it was a phagocyte, it has a special shape to allow it to engulf and digest phagocytes. If it was a B lymphocyte, it would be able to produce antibodies. Again, any type of lymphocyte would have special receptors specific to the antigen on its surface. The nervous cell at the bottom is specialised because it has that special shape. It's able to generate electrical impulses, and it has neurotransmitters stored in vesicles at the end of the axon. So these are all specialised cells. Stem cell changes to become one of those specialised cell types. Now that's a permanent change. It's one direction, it doesn't change back. And that process is called differentiation. So differentiation is when your unspecialised stem cell changes into a specialised cell type. Now if you're asked to explain the biology behind that, the cell begins to express certain genes to produce the proteins which are characteristic of that cell type. So remember, every somatic cell has all 46 chromosomes, so every cell has all of the genes, but most of these genes are turned off. When the stem cell starts to turn into the specialised cell, it starts turning off these genes that aren't required, but importantly, it starts to express the genes which are going to lead to the production of the proteins which make it into that type of cell. Now, the muscle cells are first column. It has gene A expressed or turned on, gene B is turned off, and gene C is turned on. So if A and C are both turned on, it starts making the proteins which turn the unspecialised cell into the shape of the muscle cell. The second example is the neuron. It has genes A and C switched off, but if it's expressing gene B, it starts making the proteins that gene B codes for, and that turns it into a neuron cell. Now, a skin cell also has gene A turned off, but it doesn't have B and it doesn't have C turned on. So it's only making the proteins that gene A codes for, and therefore it's slightly different from muscle, and it becomes a specialised skin cell or epithelial cell. So, cell differentiation is a process by which a cell expresses certain genes to produce the proteins characteristic for that cell type. Okay? And that means it can specialise and carry out a specialised function. So stem cells have two properties. On the right, first of all, a stem cell is an unspecialised cell which can differentiate into specialised cell types. Other thing that all stem cells can do is replicate themselves, and this is called self-renewal, so you don't run out of stem cells. Now, there's two types of stem cell. First of all is the tissue stem cell. It's called this because it's found in various tissues around the human body. And that's important because over time you need to grow or you need to repair. Now, that's the job of the tissue stem cells. If you damage your tissue, the cells that are damaged are dead. They can't be repaired. Remember that National 5? However, the tissue itself can be repaired because the tissue stem cells will divide to make new replacement cells, and that replaces the damaged cells and fixes the tissue. Now, tissue stem cells are called multipotent. Now, potent because it talks about their potency, like how many different cell types can it turn into. All tissue stem cells are multipotent as they can only differentiate into the types of cell that are associated with or found in that particular tissue. So if it's in the muscle tissue stem cells can create any cell that you'll find in the muscle, but it can't create something else like an eye cell or a red blood cell or something different like that. In your bone marrow, you have a different type of tissue stem cells, and those tissue stem cells can only change into types of blood cell because that's the cell associated with the bone marrow. So it can make B and T lymphocytes, platelets, and red blood cells. The lining of your small intestine has tissue stem cells, and those tissue stem cells are multipotent, meaning that those tissue stem cells can only differentiate into the types of cell that you would find inside the small intestine. Okay, so I think multipotent starts with an M, so you can maybe think about muscle as a type of tissue, as a way of linking tissue to multi, okay? But it's a limited range of cell types. Now there's an example we talked about there. The blood stem cells located in the bone marrow give rise to all your red blood cells, the platelets, your phagocytes, and your lymphocytes, okay? But it's not going to create a kidney cell or a skin cell, okay? The reason for that is some of the genes are already turned off in the multipotent tissue stem cells. So if you've already turned off the genes required to make like a kidney cell, and they're permanently turned off, that stem cell can never make the proteins required to turn into a kidney cell. However, it does have the potential to turn on any of the genes which make the proteins required for any of these blood cells, so it can make that limited range of cells. Okay, now compare it to embryonic stem cells. Now the embryo has to be able to turn into a full human. So the stem cells from the embryos have to be able to make any type of cell because they have to create the whole body. So we call these pluripotent. Now a good way to remember that is it starts with P, so just think P for pregnant. So the one to do with pregnancy is the pluripotent ones, and that's found inside the embryo. All the genes in the embryonic stem cells can be switched on or have the potential to be switched on, and that's the explanation as why it can turn into any type of cell. Now, it's really important in answers you make clear that it's got the potential to be switched on. Don't say that they are switched on. Otherwise, that stem cell will be making every protein that humans have, and that's not the case. So here you can see the zygote dividing and dividing and dividing into the early embryo. And that group of cells there is where we'll find the embryonic stem cells. And because we know they're there clustered inside the embryo, we can put a needle in and remove the embryonic stem cells. Now, why would you do that? Well, that raises some ethical issues. We want to take away these embryonic stem cells because we know they can change into any type of cell. And that means we can actually use them to develop treatments for disease and injury. So stem cell research can lead to different therapeutic treatments or understanding disease better. So that's ethical reason for doing stem cell research. However, when you remove those embryonic stem cells, that destroys the embryo. And that's an ethical concern against stem cell research. So a reason for and a reason against, and you need to know both. Therapeutic uses of stem cells. Therapeutic means a treatment, okay? Because stem cells are naturally involved in the repair of damaged organs or tissues, remember, not cells, but organs or tissues, and that happens naturally in the body, we're keen to try and use these to develop our own medical treatments, and that's what we mean by a therapeutic use. So you could talk about a corneal repair as being an example. You could use stem cells to create new corneal cells, and then transplant them into the damaged cornea to replace the damaged cells there, repairing the tissue. You could also use stem cells to create new skin cells and transplant those new skin cells onto a damaged area such as a burn, and that would cause the regeneration of the damaged skin. So again, you can repair the tissue using stem cells. These are both therapeutic treatments. If you're asked a research use, then don't talk about treatments. Research cells is when we take the actual stem cell and we use it as what we call a model cell. So that's a cell that we can do things with. Now we can use that model cell to study how diseases develop. Or we can use that model cell to test drugs, to see how the drug enters the cell, if it's uptaken well, how that actually works at molecular level. Stem cells from the embryo can self-renew in the lab in the right conditions. So it's a slight typo though. Stem cells from the embryo can self-renew in the lab in the right conditions. So stem cell research provides information on different cell processes, such as cell growth, cell differentiation, because that's what stem cells do, and gene regulation. Now, gene regulation is the turning on and off of genes. As we said in differentiation, that's what causes a cell to differentiate. So we can study that using stem cells. Again, it's your opinion, but in terms of the course, just know factually that the reason for stem cell research is because you can develop cures to diseases or treatments to disease. Ethical reason against is because removing embryonic stem cells leads to the destruction of embryos. Again, you can pause this and go through the SQA course notes, which are used to make up your marking schemes. Make sure you're able to word your answers using this language. Okay, finally, cancer. Now, cancer cells divide excessively. That's the description of what a cancer is. So cancer is when cells begin to divide excessively. Now, the reason why they do this, if you're asked to explain, is they don't respond to regulatory signals, and that results in a mass of abnormal cells, and we call that a tumour. So there's three things there. Define cancer as being when cells divide excessively. So cells divide only when they're required to, so that's controlled by these regulatory signals. If they don't respond to regulatory signals, they keep dividing and dividing and dividing, so you get that cluster of abnormal cells, a mass forms. And we define a tumour as being a mass of abnormal cells which are dividing excessively. Now, you all know that sometimes tumours can spread around the body. So how does that happen? Well, if we look at that bottom right picture, you have the tumour there and you have all the blue cancer cells compared to the normal cells either side. And one of the problem with cancer cells is they fail to attach to one another. Now, if one of the cancer cells breaks away from the tumour, it can spread around the body and the blood, and wherever it stops, it will begin to divide excessively again, forming a secondary tumour. So that's the definition of a secondary tumour and the explanation as to how it forms. Okay, that is the end of the lesson. That's SQA final notes. Make sure you go over this and get your notes made, and then try making up your own questions to quiz yourself on it. Make sure you practice retrieval to memorise this information.